All right, so let's talk proteins. We all know they're like the building blocks of life and all. But have you ever stopped to think about just how many different proteins there are out there?
Oh, it's mind-boggling. We're talking billions upon billions of unique sequences. It's like a vast, uncharted
Yeah,
universe.
and get this, we've only figured out the actual 3D structures for like a tiny fraction, something like 100,000. That's like a drop in the ocean.
It really highlights the massive challenge we've been facing for over half a century, the infamous protein folding problem.
Exactly. And for those who might not be familiar, it's basically this. You have a protein, which is essentially a long chain of amino acids, right? Like a string of beads. And this string has to fold itself up into a very specific, really intricate 3D shape to actually do its job.
It's not just random folding either. The shape determines everything about how a protein functions, what other molecules it interacts with, what chemical reactions it catalyzes. It's the key to understanding how life works at the molecular level.
So today we're diving deep into a paper that describes a major breakthrough in this area. It's called Protein Structure Prediction. This paper introduces something called AlphaFold, a computational method that's essentially changing the game for how we predict these 3D protein structures.
Yeah, imagine going from just knowing the list of ingredients to instantly seeing the fully baked cake. That's kind of what AlphaFold is doing for protein structure prediction.
So today we're going to break down why AlphaFold is such a big deal, how it works at a high level, without getting too lost in the weeds, and then explore what this kind of technology could mean for all sorts of different scientific fields.
Think of this as your guide to understanding one of the most significant scientific advancements in recent years.
So let's start with this protein folding problem. Why has it been such a tough nut to crack? What makes it so hard to predict how a protein will fold just by looking at its amino acid sequence?
Well, imagine you have, say, even a relatively small protein with 100 amino acids. The number of possible ways that chain could fold up is astronomically huge.
It's not just about finding a shape that works, right?
Right.
It has to be the one specific shape that's the most stable and allows the protein to do its job.
Precisely. And the traditional approaches to this problem have either focused on trying to simulate all the physical interactions between the atoms in the protein.
Which sounds insanely complicated.
It is computationally incredibly demanding. Even with supercomputers, it's really difficult to accurately model all the forces at play, and even more difficult to predict which of the many possible shapes will be the most stable.
So that brings us to the other main approach the paper talks about, using evolutionary information. Can you explain that a bit more?
Sure. So the idea here is that proteins with similar amino acid sequences tend to fold into similar 3D structures.
Makes sense. Like if they're related evolutionarily, they probably share a similar blueprint.
Right, and as we've gotten more and more protein structures experimentally determined and deposited in databases like the Protein Data Bank, or PDB.
The PDB, that's like the library of known protein structures, right?
Exactly. With this growing database, combined with the advances in genomic sequencing and the power of deep learning to analyze massive data sets, these evolutionary methods have gotten pretty sophisticated. They can find subtle patterns in how amino acids change together over evolutionary time, giving us clues about which parts of the protein might be close together in 3D space.
But even with all that, there were still limitations, right? Like, what if there's no known structure for a similar protein to use as a starting point?
That was a major roadblock, and this is where AlphaFold really shines. It's able to predict structures with incredible accuracy, even for proteins that have no known structural relatives.
So AlphaFold is like, hold my beer, I got this.
Pretty much. The paper really highlights this point. They say AlphaFold can regularly predict protein structures with atomic accuracy, even in cases in which no similar structure is known. That's huge.
Yeah, that sounds like a game changer. But how do we know it actually works? How do we know these predictions are accurate?
Well, the researchers put AlphaFold through the ultimate test, a blind assessment called CASP, the Critical Assessment of Protein Structure Prediction. This is like the Olympics of protein structure prediction. Teams from around the world are given the amino acid sequences of proteins whose structures have been experimentally determined but not yet made public.
So it's a real test of how well these prediction methods work. No cheating allowed. And AlphaFold, I'm assuming, did pretty well.
Blew the competition out of the water. In CASP14, which took place back in 2020, a team called AlphaFold 2 achieved a median accuracy for the backbone of the protein of built 0.96 angstroms, RMSD 0.95.
Okay, can you break that down for us non-scientists? What does 0.96 angstroms actually mean in terms of accuracy?
So an angstrom is a unit of length, super tiny, about 1/10 billionth of a meter. To put it in perspective, the width of a carbon atom, one of the basic building blocks of molecules, is about 1.4 angstroms. So AlphaFold is predicting the positions of the atoms in the protein's backbone with an accuracy that's close to the width of a single atom. It's mind-blowingly precise.
Wow, that's some serious accuracy. And the next best method in the competition, how did that compare?
It was way off, relatively speaking. It achieved an accuracy of 2.8 angstroms.
So almost 3 times less accurate than AlphaFold. That's a huge difference.
And it wasn't just about the backbone either. AlphaFold also predicted the positions of the side chains. Those are the parts of the amino acids that stick out from the backbone and are responsible for the protein-specific interactions with remarkable accuracy. The overall all-atom accuracy was 1.5 angstroms for AlphaFold compared to 3.5 angstroms for the next best method.
So it's nailing the big picture and the fine details.
Yeah.
And this wasn't limited to small, simple proteins, right?
Right. AlphaFold even successfully predicted the structure of a massive protein with over 2,000 amino acids, one that had no known structural relatives. That was a real testament to its power and scalability.
Incredible. And the paper also mentioned something about AlphaFold being able to provide a kind of confidence score for its predictions, like how sure it is about the structure it came up with.
Yes, that's another key feature. It gives a per-residue reliability estimate called PLDDT. This score tells you which parts of the predicted structure are more likely to be accurate and which parts might be a bit more uncertain. It's like having a built-in quality control check.
So you're not just getting a structure, you're getting an idea of how much you can trust each part of it. Super helpful for researchers. And this high accuracy and reliability, it wasn't just a fluke in the CASP competition, right?
Nope. They also tested AlphaFold on structures that were deposited in the PDB after the training data cutoff date, so AlphaFold had never seen these proteins before, and the results held up. The PLDDT score accurately reflected the actual accuracy of the prediction. What's even more exciting is that this level of accuracy extends to completely new protein fold structures that have never been seen before.
Okay, that's huge. Think about it. We're talking about predicting the shape of something we've never encountered before, just based on its amino acid sequence. That's like looking at a list of ingredients and being able to predict not just what the dish will look like, but also how it will taste and smell. It opens up a whole world of possibilities for discovering new biological mechanisms.
Absolutely, and it's a key point highlighted in the supplementary materials of the paper.
Okay, so we've established that AlphaFold is a major breakthrough. But how does it actually work? What's the secret sauce that allows it to make these incredibly accurate predictions?
So, at its core, AlphaFold uses these really complex neural network architectures and some clever training techniques. These methods incorporate information from evolutionary relationships, fundamental physical principles, and geometric constraints. One of the key aspects is how it uses multiple sequence alignments, or MSAs, of evolutionarily related proteins.
So it's looking at the protein's family tree, seeing how it's changed over time.
Exactly, and it combines that information with features that describe the pairwise relationships between the amino acids in the sequence. Think of this as looking at which amino acids tend to change together during evolution, hinting that they might be close together in the 3D structure. All of this gets fed into a specialized module called the evoformer.
The evoformer, that sounds kind of like a transformer from those sci-fi movies.
It's a fitting name, actually. Think of the evoformer as the brain of AlphaFold. It takes all this information about evolutionary relationships and pairwise features and starts to form hypotheses about the protein's 3D structure. It does this through a series of sophisticated processing steps, refining the information as it goes along. What's remarkable is that these initial structural ideas start to emerge really early on in this process and get continuously refined and improved as the information flows through the network.
So it's not like it suddenly spits out a structure at the end. It's more like a gradual process of building and refining.
Precisely. And one of the key innovations here is how information is exchanged between the MSA or representation and the representation of the pairwise features. This allows AlphaFold to reason about evolutionary relationships and spatial relationships simultaneously.
It's almost like it's thinking, okay, these amino acids always change together, so they must be close in space. But this other pair rarely changes, so they might be further apart.
That's a good way to think about it. And then comes the structure module, where things get really interesting.
This is where the actual 3D shape starts to take form, right?
Exactly. The structure module takes the highly processed information from the evoformer, particularly the refined pairwise relationships and the original amino acid sequence, and uses that to build a 3D model. It starts with what's called a residue gas. Imagine each amino acid just floating around in space with its own independent rotation and translation.
So it's like a chaotic cloud of amino acids at the beginning.
Yes, but then, through a series of computational steps, it rapidly assembles this cloud into a coherent, accurate 3D structure. And one of the really clever things it does is temporarily break the constraint that the amino acids have to be linked together in a chain.
Wait, so it breaks the chain apart and then puts it back together. Why does it do that?
It's a brilliant strategy. By temporarily breaking the chain, it can make local adjustments and refine the structure of complex loops and twists that would be really hard to model if it had to keep the chain connected the whole time.
Makes sense. It's like if you're trying to build a complex Lego structure, sometimes you need to detach a few pieces to get the angles just right.
Exactly. And then there's another important component called invariant point attention, or IPA for short.
IPA, that sounds like a new type of beer.
Right. It's a specialized type of attention mechanism that takes into account 3D geometry. Think of it as a way for AlphaFold to fine-tune the local arrangements of the protein's building blocks without messing up the overall structure.
So it's like making small adjustments, but in a way that respects the 3D geometry of the protein.
Exactly. It focuses its attention on spatially close parts of the protein, ensuring that these local adjustments don't create any impossible configurations. And all of this gets refined further through a process called recycling.
Recycling, so like putting the protein structure through the same process multiple times.
Exactly. The predicted structure is fed back into the same modules, the evoformer and the structure module, multiple times. Each time, it gets a little bit more accurate, like polishing a rough gem until it shines. Finally, there are small specialized networks at the end that predict the angles of the side chains, the per-residue confidence score, the PLDDT, and the overall similarity of the predicted structure to the true structure, the PTM.
And all this computational power is fueled by an enormous amount of data, right?
Absolutely. AlphaFold was trained on a massive data set of experimentally determined protein structures from the PDB. But they also used a really clever technique called noisy student self-distillation to boost its performance even further.
Noisy student self-distillation. That sounds intense.
It's a fancy name, but the concept is actually pretty straightforward. Imagine you have a really good student who's learned a lot from their teacher. Now you give this student a bunch of new problems to solve on their own, and they come up with some pretty good solutions even though they haven't seen these exact problems before.
So they're applying what they've learned to new situations.
Precisely. And then you take these student-generated solutions and use them to teach a new student who might be even better than the first one.
So it's like the knowledge is getting passed down and refined through generations of students.
That's a great analogy. In AlphaFold's case, the first student was an already trained version of the network. It predicted structures for a huge data set of unlabeled protein sequences, and then those predicted structures, after being filtered for high confidence, were used to retrain the network, making it even more accurate.
Brilliant. They're basically leveraging the vast amount of protein sequence data out there, even if we don't have the experimental structures for all those proteins.
It's a clever way to make the most of all the information we have. They also used a technique borrowed from language processing called BERT-style training.
BERT, like the Sesame Street character.
It's actually a powerful language model. In this context, it means they would randomly mask or mutate parts of the input MSA and have the network try to predict the missing information. This helps the network learn the underlying patterns and evolutionary relationships within protein families.
So it's like giving the network a puzzle to solve. Fill in the missing pieces based on what you know about protein evolution.
Exactly. And all of this training and optimization has resulted in a network that can predict protein structures with amazing accuracy. And what's really fascinating is that the researchers have been able to peek inside the network and see how it actually arrives at its predictions.
Wait, so they can see what's going on inside the black box?
To some extent, yes. They did experiments where they looked at the intermediate structure predictions at different stages within the network. And what they found was that the improvements in the structure were surprisingly smooth and continuous. It makes small, incremental adjustments, getting a little bit closer to the correct structure with each step. And the way it solves different proteins is really interesting.
So it's not a one-size-fits-all approach.
No. For some proteins, the correct structure emerges pretty quickly, while for others, the network might have to explore different structural possibilities and rearrange large sections of the protein over many computational layers before it arrives at the right answer. It's almost like you can see the network thinking as it works through the problem.
Like some proteins are easy puzzles and some are super complex.
Right. And they also did ablation studies where they systematically removed different components of AlphaFold to see how much each part contributed to its overall accuracy. These studies clearly demonstrated the importance of elements like IPA and recycling.
So even with all this incredible capability, are there still limitations to what AlphaFold can do?
Of course. No system is perfect. One of the main limitations is that its accuracy is strongly tied to the depth of the multiple sequence alignment you give it as input.
Meaning, how many evolutionarily related sequences are available for that protein?
Exactly. If you only have a few related sequences, say less than 30, the accuracy of the prediction can drop significantly. But once you have over 100 sequences, the gains in accuracy start to level off.
So it still relies on having a good amount of evolutionary information to work with.
Right. Another limitation is with proteins that don't have a lot of interactions within their own chain, but interact extensively with other molecules. These are often found in large protein complexes.
So it's better at predicting individual protein structures than those that are part of a larger assembly.
For now, yes. The current version of AlphaFold focuses on predicting the structure of single protein chains, but it actually does a pretty good job with proteins that form multiple copies of themselves, like homers. The paper even shows examples of it accurately predicting the structures of complex, intertwined homomers.
That's pretty impressive. So it can handle some level of complexity in those cases.
Yes, and the researchers are optimistic that the core principles behind AlphaFold can be extended in the future to predict the structures of full heterocomplexes, those made up of different interacting chains. That would be another massive leap forward.
It feels like we've entered a new era of structural biology. And the paper already hints at some of the potential applications and impacts of this technology.
Absolutely. AlphaFold really is a fusion of traditional bioinformatics knowledge and cutting-edge deep learning. The fact that it learns directly from the vast amount of structural data in the PDB, without relying on a lot of pre-programmed rules, allows it to handle a huge diversity of protein structures and experimental conditions. It can even make accurate predictions for proteins where the experimental conditions for folding weren't fully defined in the PDB entry, like cases where the presence of certain cofactors or binding partners is unknown.
That's pretty amazing. It's like it's learned the general principles of protein folding so well that it can make accurate predictions even when some information is missing.
Exactly. And this is already proving to be incredibly useful for scientists who are trying to determine protein structures experimentally.
The paper mentions things like molecular replacement and interpreting cryo-EM maps. Can you explain what that means for those of us who aren't structural biologists?
Sure. So there are various experimental techniques for determining protein structures, like X-ray crystallography and cryo-electron microscopy, cryo-EM. These techniques involve collecting data about the protein and then using computational methods to build a model of its structure.
So it's kind of like taking a bunch of blurry photos of the protein from different angles and then trying to piece together a 3D image.
That's a good analogy, and AlphaFold can provide incredibly accurate starting models for these computational methods, which can significantly speed up the process of determining the structure.
So it's like giving the scientists a head start in solving the puzzle.
Exactly. And the speed of AlphaFold's predictions is remarkable. It can generate a structure in minutes to hours, depending on the size of the protein, using a standard graphics processing unit, GPU.
So we've gone from potentially years of experimental work to get a single protein structure to being able to predict structures in a matter of hours. That's a game changer.
It really is. And this speed has opened up the possibility of predicting the structures for entire proteomes, all the proteins expressed by an organism. In fact, the researchers have already done this for the human proteome, publishing those results in a companion paper.
So we're talking about having a structural map for almost every protein in the human body. That's mind-blowing.
And this is just the beginning. AlphaFold, combined with all the genomic and sequence data we already have, has the potential to revolutionize structural biology and accelerate discoveries in countless areas of research.
So to sum it up for our listeners, AlphaFold is a truly monumental advancement. We've been wrestling with this protein folding problem for over 50 years, and now we have a tool that can accurately and reliably predict protein structures, giving us an unparalleled ability to understand the very building blocks of life.
It's like having a superpower. We can now look at a protein's amino acid sequence and visualize its 3D shape, unlocking a wealth of information about its function and how it interacts with other molecules.
And this isn't just some abstract scientific achievement. It has the potential to revolutionize fields like drug discovery, material science, biotechnology, you name it. Imagine being able to design new drugs that precisely target specific proteins or create novel proteins with tailored properties for all sorts of applications.
The possibilities are really endless.
And we want to leave you with a thought to ponder. How might this newfound ability to predict protein structures impact areas of science that you're personally interested in? What new questions can we now ask? What doors have been opened that were previously closed? AlphaFold has given us a powerful new lens through which to view the world of proteins, and the discoveries we make with it are sure to be remarkable.
And for those who want to delve into the technical nitty-gritty of how AlphaFold works, we encourage you to check out the source material, the paper proteinstructureprediction.pdf. It's a fascinating read.
Thanks for joining us on this deep dive into one of the most exciting breakthroughs in recent scientific history. It's an incredible time to be following the world of science, and we can't wait to see what unfolds next.
Absolutely. Until next time.